Our last presenter of the day is Dr. John Morris from the Scott Lowe Lab in Cancer Biology and Genetics, and he's going to talk to us about Mr. P53, the Cell Guidance Counselor.
So we have just experienced a very important and exceptionally frustrating anniversary in cancer cell biology. 30 years ago, mutations in the gene P53 were specifically found in a patient's tumor and not in their normal cells. And right after this finding, there were two patterns that became extremely evident. One, these mutations eliminate the wild-type function, the normal function of the P53 protein produced by the gene. And these mutations occur in this gene more than any other gene in human cancer. So since then, for 30 years and about 96,000 publications, we've been trying to determine how we can weaponize these two patterns to be able to treat P53 deficient tumors. And given that we know how P53 works, it is a gene that turns other genes on and off in a very unique way, and we know that it's the most commonly mutated gene in human cancer, we should be able to weaponize, and we should all be able to go home. Unfortunately, our models of how P53 actually affects the ability of cells to become cancer haven't been predictive. So P53 is a tumor suppressor. It's a gene, when it's lost, predisposes cells to becoming tumors. And as I said, it turns genes on and off in a very unique way, in response to stress rather than specific signals. And we've actually found thousands of genes that P53 can turn on and off. But not all of these genes are mutated or lost in human cancer, and oftentimes these genes can actually be required for a cancer to form. So I propose that we need to think about P53 not as what I've been doing for the past minute and 30 seconds, which is giving you universal facts about a very important protein, and acting as a teacher, where it will respond to specific stresses and give rise to just very specific molecular outputs. But rather, think about it as a cellular guidance counselor, where P53 will respond to contextual stresses to be able to produce a number of cell fates by turning on a number of genes that might be involved in the prevention of cancer. And this is where my postdoctoral arrogance comes in, and I think I can contribute something more than those other 90,000 publications. So rather than taking a normal cell and giving it a stress that might be involved in the production of cancer, we take cancer cells that are experiencing the specific genetic events that drive tumorigenesis, and we restore P53 there. By studying the cell fates and going back to the genes that P53 can turn on and off, we can actually find specific patterns that we can restore in P53 deficient tumors to be able to get anti-tumor effects. So by thinking more broadly, as a cell guidance counselor, we can make the next anniversary of P53 research a little bit more happy. Thank you.